Let's just wait 1 minute.
Yeah.
Recording stopped.
Say hello to everyone. Patrick, everyone is hearing us, right?
I think so.
Yes, we can hear you. Thanks.
Okay, great. Thanks a lot.
But I think it's worth waiting a few minutes because people are still signing in.
Yeah. And Steve Aoka is also joining. And
Great.
Musa, yeah. Okay. Thanks a lot, Libby. So hope everyone is well. Patrick, Steve has some difficulties with the link.
Okay.
If you can send him again by email.
Okay, I'll do so right now.
I think we can start now.
Okay, so hello everyone. Hope everyone
Recording in progress.
is doing well. So it's a pleasure for us. WHO, Africa CDC, and INRS on the behalf of the CORC to have this meeting. We will try to present to you all where we are today with the research around the epidemic currently occurring in DRC and Uganda. So I will maybe before any other thing give the floor to Vazi Muthi to present an introduction on the behalf of WHO. So thanks a lot, Vazi, for your talk.
Thank you so much, Yazdan. So my name is Vasi Morthy. I'm the head of the R&D Blueprint at WHO. Good morning, good afternoon. I'm speaking because both Sylvie Briand, our chief scientist, and Chikwe Ihekwazu, our executive director, are in urgent meetings. So they asked me to open on their behalf and send you their regards. It's been almost 2 months since our first emergency scientific consultation on Bundibugyo that was co-hosted, as with this one, by WHO, ANRS, the lead Filovirus CORC, and Africa CDC. I'm really encouraged by the remarkable progress that's been achieved with an urgency, with coordination of research integrated into the response. Over the past 2 months, I've seen the leadership by DRC and Ugandan scientists and public health agencies. I have witnessed and sought to enable scientific collaboration in the service of bringing advances to communities that can save lives and help contain this outbreak under extremely challenging circumstances. I visited Bunia and Kinshasa to better understand what WHO can do to support the best research integrated into the response and to meet the research leadership of INRB, the partners, INSP, the DRC Ministry of Health. Your collective efforts demonstrate what is possible when scientific collaboration is mobilized early in an outbreak. We're already seeing important milestones with the support and leadership of the authorities in Uganda and DRC. Just to mention briefly a couple of them, the Partners Therapeutics trial has begun enrolling patients with INRB, WHO, Africa CDC, Oxford, ITM, ALIMA, MSF, Samaritan's Purse, IMC, all working together, evaluating promising treatment options with the aim of hopefully making proven, safe and effective options available beyond the trial if results are promising. This week, led by INRB, ANRS and ALIMA with support from Africa CDC and others, the first post-exposure prophylaxis trial using oral obeldesivir was launched among high-risk contacts. If this is successful, this could fundamentally change how we interrupt transmission and protect those most at risk. This oral prophylaxis agent this is really a new era for Ebola outbreak can take us forward. And earlier this week, the world's first phase 1 clinical trial of a Bundibugyo specific vaccine candidate, the ChAdOx1 BDBV, was initiated by the University of Oxford, with Moderna also progressing very quickly. And this happened as WHO released the target product profile for Bundibugyo vaccines. So for me, this all shows what we can do together to bring science in service of communities. I see and I expect no competition, only collaborative service, and I want to acknowledge the very close partnership with Africa CDC, INRB, CEPI, who we work very closely together with, our CORC ANRS. This is exactly the kind of coordinated research ecosystem that the R&D blueprint was established to enable, but at the same time, we should recognize that there are gaps. We don't have counterparts to CEPI, to Gavi and UNICEF for diagnostics and therapeutics in the same way. And so we're calling on partners to ensure adequate post-trial access, acknowledging the really important work of Unitaid here. Clinical trials are clearly just one part of research, and we absolutely need to focus on the non-medical countermeasures research and the community engagement work. And throughout this whole process, community engagement is completely fundamental. And I didn't want us to squeeze this into one session of one meeting. So we agreed that we will have a separate meeting on the equally important areas of social behavioral science, which underpins all of this, and other non-MCM research. The progress that we'll hear today belongs to all of the partners that are working collaboratively together. And this isn't in the talking points that I was given, but and WHO doesn't normally do this, but I think we should really acknowledge the private sector entities that have been working. We depend on the private sector. And so MapBio, Gilead, Moderna, Oxford with Serum Institute of India, and many others have stepped forward, and we really thank them for answering the call. So with this, I wish you a productive meeting, and I look forward to the discussions ahead, and hand back to Yazdan.
Thanks a lot, Vazi, and thanks for this word. I wanted to say to those who are French speaking that there is a button on your screen, plus. If you push to that button, and Vincent is going to write it in the chat, you will have the traduction in French if you wish. So just wanted to mention this, and thanks again, Vazi, for your speech and for all you are doing since the beginning, of course. And now I wanted to give the floor to Dr. Musoka on the behalf of Africa CDC. We are doing this work really all together with Africa CDC, with WHO, and it's really important. So thanks a lot. And the floor is yours, Dr. Musoka.
Good afternoon, everyone. I hope you can hear me.
Yes, we can.
Easy, 30 kilometers from Bonia to celebrate the good news for tens of hours I've been discharged today, and so the signal will be weak. But to just leverage upon what Vasi said and Yazdan has been putting, you see the beautiful tapestry of different panels I've worked here. 3 days ago, we were here to evaluate the site in Nizi and see how we will start a trial for the partners trial. What is nice was that we're here with Diallo from WHO, me, the national gypsy was here from ENRB, and we had Lawrence and the team from ITM walking through the entire infrastructure to see where we could best place the treatment center in the spirit of collaboration. So that's collaboration in action and not in speech. I've come on behalf of the DG, Jean Kaseya, leadership of Africa CDC, together with Placide, to say they send the greatest greetings and the collaboration happening on the field and every day. We drove today 30 kilometers from Bonia, not only to attend the survival discharge, but also to start to work with the local teams and see how we can quickly set up a decide for Ebola that could be moving quickly, as we discussed with Yap and Placide, that one of the things we could do wherever there was a new hotspot, we could quickly and erect a structure to support this. So from being on the ground at the operational level, you know, what was initiating the trial in Rampara with WHO, Africa CDC, the team from Alima, ENRS to go through the simulation. We've seen real collaboration in research. So, and what has done for us, we leverage our own individual strength, and we are able to accelerate the process. What it was a few days ago, we met with the team from MSF Epizone to sit together and derive a vaccine protocol. You know, it went very fast. We had two different protocols, but we agreed we merge the protocol. We go one correlation. You know, Vasi spoke about the non-pharmaceutical approach. I'm working right now with Renee to look at how we can bring different groups together to create one big protocol in epidemiological transmission dynamics. Yesterday, we held a meeting with UK FCDO and other partners from the different social scientists from the Epi board and from the partners, try to see how we can launch the social behavior study and also include the perception of the communities to new products and trials, engaging with CEPI and other partners on an everyday basis. For those of us on the ground, we see close relationship, openness as we collaborate, we brainstorm, we visit the site, we talk to a representative from all of the different partners. It is really working, and I can speak from the ground that all of your commitment, support, and resources is really translating into actual collaboration that I think if we succeed tomorrow is going to be because of one thing. We've seen a new dawn in collaboration and partnership, openness, and leveraging each other's strength. So again, thank you so much. My colleague Plessy will speak later on from some of the studies, but it's been going great, and I can assure you, being here in Nazi, that with the leadership of the local scientists, Africa, WHO, ENRB, and all the partners, including our funders, our private group like FHR 360, they're all doing an excellent job. We thank God for Amatu has been coming to work with us to be able to push this collaboration. So together we win, together we succeed. Thank you very much.
Yeah, thanks a lot, Dr. Musoka, and also thanks a lot really for everything that you are doing. Huge amount of work, and we are all recognizing what you're doing, and thanks you a lot. I just wanted to put you here now, the agenda, so that you know where we go. We try to keep this in 2 hours. And as it was said by Vazi, here the idea is also to tell you what has progressed, in particular regarding some clinical trials on treatment, but also on vaccine, on post-exposure prophylaxis. Also present you some where we are with diagnosis, an overview of research priorities. And as Vazi said in the beginning, and also Dr. Muzoka, it was hard to do this, all what has been done in 2 hours. So we are going to have another meeting next week to try to follow on this and tell you also what has been doing in other areas than what we're going to present here. So virus modeling, a little bit more about the natural history in particular. So this is the plan for today. We wanted to actually ask Dr. Auka, that has been, I think, 2 or 3 days ago, appointed by the President of Republic of DRC as the outbreak manager of the DRC for this outbreak, to give some opening remarks. I think that he's still not there because we have some connection issues, but he will join. What I propose is that we start with Marie-Roseline Belizer from WHO, if you agree, that I can see she's here, and then who on the epidemiological situation today. And then we will follow up with Steve that I'm going to bring in. Marie-Roseline, thanks for your presence.
My pleasure. Can you see my screen? I'm putting it in a presentation mode.
Yes, it's perfect. Just on, if we can have it on, yeah, it is not on presentation mode, but I think we can.
It is not yet?
We can see it, but it is not on presentation mode. But yeah, that is exactly. If you do that, it is perfect, Marie-José. Thank you.
Wonderful. Okay, thank you so much, Yatan. So good morning, everyone, or good evening. So as you say, my name is Marie-Rosine, but I will be using the presentation that Etienne, the AP lead in the program I'm managing, will be presenting soon in the joint continental IMST. So as you know, the outbreak will continue to evolve. So we are at 2 months of the declaration, 2 months and 2 days for today, that 62 days. And as you can see now, the summary of the key figures that we have. So far in the Democratic Republic of the Congo, the outbreak continues to evolve. As of 15th of July, we have 2,124 confirmed cases that has been registered in this outbreak, with 823 confirmed deaths among the confirmed cases. That's really put higher the case of fatality rate at 39%. But we also have good news, because we have 390 patients that has been recovered from the disease so far. So when we look at now at Uganda, so Uganda yesterday discharged the last patient that was in the Ebola treatment center. So they started the countdown with 20 confirmed cases and 2 confirmed deaths. That was a 10% of case fatality rate, and they have 18 recoveries, and the last one was yesterday. So today is the first day of the 42-day countdown for Uganda. So this is an epidemiological summary. As you look at, as 15th of July, as you say, we have 20 confirmed cases in Uganda, and 15 were imported, and 5 are Ugandan that were really contact of those that has been imported. So the comparison, we cannot make a comparison between those 2 outbreaks, but we have to only say that they are different type of outbreak and managing differently. So in Uganda, the contact tracing was 100%. So they have 831 contact that has been listed, and all of them has been followed. And during the period of the response, they analyzed 2,482 samples that has been tested. That means it was very a large operation to that. So now when we look at the Democratic Republic of the Congo, the latest data we have, we see that our cases continue to increase. And yesterday we have 51 new confirmed cases, but the most alarming indicators is that we have 32 deaths within those confirmed cases. That is really an indicator we care about, because it's telling us how access to healthcare is the community is accessing, is the community also accept the response and are going to healthcare earlier before the disease becomes severe. So I will not go through, the breakdown of those 51 cases, but what I wanted to see is that we still have 10 of those health zones that continue to be with the highest number of cases. So now it is the geographical distribution of the confirmed cases in the DRC. So far the outbreak has been reported in 5 provinces and in 46 health zones. And the last health zone that has been affected is the Mahagi health zone. The Mahagi health zone is bordering also with Uganda, like Awu is also bordering with Uganda. So we can see that in Uganda, the outbreak, they started their countdown with 1 district affected. So the trend in confirmed cases, as you see in the Democratic Republic of the Congo, continue to increase. The outbreak continues the ascending trend we continue to see in DRC. So the trend also in confirmed cases are the same in DRC. So we have the epidemic curve by week, as we have more than 2,000 cases. It will be very difficult to have it by day. But however, you can see that in the week 25, the week 26 and 27, so it was the week where we have the highest number of cases registered in this outbreak. So if you look at, you can see, oh, maybe there is a descending trend, but it is very early to say that. So the trend will continue to register a very high number of cases every day. So this is the projection, the short-term projection that has been done. So as you see with the projection, we continue to have an ascending curve. So the projection has been made with a confidence interval of 2,130 to 2,309. And we see that the projection was saying that by 15th of July, we will have this number, 2,210 cases. And I think we are very close. We are already at that, very close to it, because we already have 2,000 and more than 2,000 cases. So now this is the distribution by cumulative confirmed cases by health zone and by provinces. As I mentioned, we have 10 health zones that are maintaining the highest cumulative number of cases. And those are continue to be Bunia. So far, we have 570 cases. We continue to also have Rampala with 418 cases and Mongwalu. And now we have Nizi that has been also added. And you know that the particularity of Nizi is that it is a place we also have an internal displaced population who are also affected in the Ebola. And then we have Nyankonde, Lita, and Mongalia that are also affected. So this is I can say when you can see the curve by health zone. So you can see that the pattern is totally different in each health zone. So in green, you have the confirmed case that are alive, and in red, you have the death. So as you see, for example, in Bunia, you have the most concentrated death than in Rampala. So however, as I mentioned, the pattern of distribution of the cases when you look at the epidemic curve is totally different from a health zone to another. So when we look at also, this is the same one. So when we look at those, the crude case fatality ratio, so those are deep analysis that the team is conducted in order to see where is the case fatality rate more by inside the health zone, by health, I can say, health area. So when you look at, because even though we have a Bunia, so but we look at deeper in Bunia, you will see that not all the health area in the health zone are affected equally. So this is also the same curve, but by community death and also by the death in ETU. It is a very important data we want to share here, because this is where all the researchers will have really that I can say that to decrease this indicator of death inside and also in the community. So far, when you look at more deaths are still coming from the community. That means, for example, if you look at, for example, in 7th of July, it is where we have 84% of the deaths that were registered that day came from the community. So it's also, inform a lot of indicators that we will not be discussing here for time. So now when we look at the third epidemiological data, which is the age and sex and distribution, we can see that more cases are concentrated among the group age of 20 to 48 years old. And also the deaths are also concentrated, more than half of the deaths are also concentrated into the same age group. But you can also see that women are more affected than men during that outbreak. So we also have the case fatality ratio by lower age group. For example, when we look at the children under 1 year old, we can see that the case fatality rate is very high. It is 63% in those children, and it's decreasing when you look at 1 to 4 years old and less than 5 to 9, and also, and you can see what is increasing in adults that are a superior at 16 years old. So it's very important to see, even though we have more women affected by the outbreak, however, we have more deaths registered in male. Okay, so I will stop there. I think this is the last that epidemiological data that I wanted to share. So thank you so much. Over.
Yeah, thanks a lot. I think these are very important data. You know, what we are going to do probably is to have maybe the partner study first before having some question, or because the Q&A is just after after the partners study. So we will do that and then come back to your question, because I think there are many and very important data here to be discussed, and I'm sure there will be a lot of questions. So thanks a lot. In the next, Steve Auka is going to come and relieve his remarks maybe in the beginning of question and answers. We had put a session on the clinical characteristic of patients infected with Bundibugyo virus, presented by Pierre Alicami, actually, who is from INSP. Actually, what I talked with Placide, it seems that Pierre was, you know, that is the issue when you have people who are on the field. Pierre is on the field with the governance today. We were not sure that he will manage and if he will be there. What he wanted to present was published in New England Journal of Medicine, and I know that editors and Lindsay Biden of New England Journal of Medicine are here. So if we don't manage to present the results, you can find it the paper, and we will send it to you if he doesn't make it. But for now, I propose that we move actually to next topic, which is actually an update on the partner study on treatment. It will be done by Gypsy Yongo from INRB, who is present here, and Amanda Rojek. So I don't know who is going to present, but both are present. And also, even before they start, just thank them for all the efforts for getting this started. Over to you, Gypsy, who is one of our champions, and Amanda.
Thank you very much, Dr. Yazdan, for the floor. So my name is Gypsy Yango. I'm a medical doctor. I will present on behalf of the partner consortium. Yeah, so rapidly about the key information regarding the partner trial. So the sponsor is WHO and also the country. So the design for this trial is an open label adapted platform randomized controlled trial with the factorial design. So our population is patient with the laboratory confirmed acute Filovirus disease, included also Bundibugyo. So in terms of a key eligibility, the eligible participant here is positive participants from the RT-PCR, and also participants are admitted to the Filovirus treatment with no contraindication to the study drugs, and also with not previously enrolled or not prior antiviral and also monoclonal antibody treatment during the current infection. But we also have a special population, especially in this trial, and the special population included children, pregnant women, and also breastfeeding women, and participants in another trial, like vaccine study, like also Ebola. So regarding the current comparison for Bundibugyo, so we have MBP 134 versus not MBP, and also remdesivir versus not remdesivir, and the primary outcome of this study is all-cause mortality at 28 days following the randomization. But we also have a secondary outcome, which is the time to viral clearance. And another outcome, especially for this study, is the viral load, the organ dysfunction progression, safety outcome, pregnancy, and also fetal outcome. Related to the safety reporting, this study is mainly focused on the adverse event and also the treatment related SIA and also the SUSA. So really important to know that the SIA related to this trial should be normally based on the treatment, but not due to the Filovirus disease and not also related to the pregnancy outcomes. So before this outbreak, the trial protocol and also the trial management was developed and finalized. It was in 2024. So the statistical analysis plan was also developed, all the different procedures, but we developed also a core protocol. And the core protocol with the different annex from countries and annex also for virus. The first country was Rwanda. And also for the virus, it was Ebola, so Zaire, Sudan, and also Marburg. At the same time, the team training and the capacity building was done. The community advisory panel also established. And we put in place the electrolink trial system like the electronic data capture for the ECRF and also the randomized platform. And the study was registered and all the regulatory approval from WHO was obtained at this time. And I can just confirm that the activation, one site, so in Rwanda, the activation and enrollment of the first study was within 3 weeks after the Rwanda Marburg outbreak. What about the current situation? So actually, as I said, the protocol is already in place. And as you can see, we had also Uganda and DRC. And especially for the virus annex, we had the Bundibugyo annex. So actually, we have Ebola, Sudan, and also Marburg, and the Bundibugyo substance that is already incorporated to our protocol. In terms of current progress, so I will go more maybe in the trial implementation. So for the trial implementation, all the regulatory and the ethical approval was already obtained. And the trial is actually successfully activated in one site in the Bunia. And we are still continues with the participant recruitment. So the participant recruitment is actually ongoing, and we are conducted also a high quality data collection. But at the moment, we think that for the future, 2 additional sites is expected to be open shortly. And the first monitoring visit is actually underway. But in terms of collaboration and also partnership, we are developing some agreement from all the 4 NGO in place in the DRC. And for the future also, we plan to expand the platform into Europe because this experience is really important example for of equity in terms of global health. So here you can see the different consortium, the consortium partners. We have INOB, INSP, University of Oxford, ITM, Ghana University, Mbarara University from Uganda, Ivory, Uganda, Ministry of Health of Rwanda, and with the support of Africa CDC, and all sponsored by the WHO. You can, if you click to this link or the QR code, we will get more information about the partner trial. And yeah. So that's all for my presentation. Thank you. I'm really open to respond to your questions.
So thanks a lot, Gypsy. Now we are going to open to questions to actually Marie and to Gypsy, Amanda, and I just found out that Placide is hidden behind Gypsy. So they're together, because I was just saying, where is Placide? But so Placide can respond to. But can you just maybe upgrade Placide? But they are here. And also Steve Aoka joined us, but we do the question on his request, and he will talk after the questions so that we don't actually cut the continuum of the talk. So so basically, maybe Marie, there is a question for you first on whether there are data on healthcare workers.
Yes, thank you for the question. Yes, we do have data on healthcare workers. I think we summarize the data, we present all the data that we have.
Yeah, of course.
Yes, we do. For example, in Uganda, we have 5 infected cases that were national, and 4 of them are healthcare workers. And in DRC, I think the latest that I have in healthcare workers, we have 112 who have been affected, and more than 30 have died so far. Thank you. Over.
Okay, thanks a lot. There is another question, because maybe it was not clear, so maybe Gypsy and Amanda and Placide, you can respond. I think that not everyone got the fact that it is a factorial design, and some people could get also the monoclonal and remdesivir together. So maybe good to maybe comment on that.
Yeah, I can comment on that to begin with. So we have 2 arms open in the moment. This is a kind of partially factorial trial design. So the first arm is the monoclonal arm, and the current comparison is MBP134 versus standard of care without a monoclonal. And then the second arm that is active is a small molecule antiviral arm, and that's remdesivir versus best standard care alone. There's a host-directed arm that's not open at the moment. What that means in terms of the patient allocation is approximately 1/4 of patients will receive the monoclonal alone, 1/4 will receive remdesivir alone, 1/4 of patients will receive the combination of both of those treatments, and 1/4 of patients will receive optimized supportive care with no additional potential treatment.
Thanks a lot. There is another question on whether you consider the rate of viral decline as a more robust measure of viral control, less affected by the initial viral level, I imagine, when compared to undetectable viral load. So I don't know if you have any answers to that. And
Yeah, we do viral load kinetics. It's obviously not our primary outcome. So primary outcome mortality at 28 days, and secondary outcomes organ dysfunction, so liver and kidney, and then also viral load kinetics.
Thanks a lot. So the question is whether the combination arm is not yet open, and why you haven't opened it yet?
It's open. So it's not a combination arm, it's a factorial trial design. So, but there are patients who will be receiving both the monoclonal.
Okay.
Yeah.
I think that is clear. There's also a comment that says that it is also great that children and pregnant women are involved. How will other ethical and regulatory bodies in Africa learn from the ethical progress that you have done on approval and monitoring of this trial from DRC? So that is, I think, a comment rather than a question. There are questions, and I think this is more for Gypsy and Placide. Are there challenges related to quarantine and isolation? Gypsy?
The challenge.
So this is Placide. I will just say everyone because I know Placide's voice. This is Placide.
Yeah. Placide, yeah.
Please, Placide, go on.
Yeah, I don't see any challenges regarding the isolation. I don't know if it's the question.
I don't know. It is, yeah, it is. I think what does it mean, and what the person can maybe specify more is that given that you have people in isolation, whether it's a problem when you enroll in the clinical trials.
No, because as you know, for the clinical trial, all the patients are isolated and they are already confirmed, so they are in the red zones, and they will be there until all their tests will become negative. So they are getting care, and so the study is a part of the care that they are getting. The difference here is that they will need to provide a consent and then to be part of the study. But we don't see any issue for the moment.
Okay, thanks a lot, Placide. And then Marie, there is a question for you, which was my question. You know that I don't have the right to ask myself question, but I think one data that you showed that it's very important is the large amount of community deaths, up to 84% on July 6th. Do you have then the people, of course, when you say that, then link the number of deaths in the community and the chain transmission? So given that you have those in the community, do you have data on I mean, what are your comments regarding that?
Right. Thank you for the question. I think other colleagues in the call can complete. I think we are all in the field, Placide, Auka, everyone can complete. So indeed, we continue to note a high number of community deaths, and we also continue to note some cases that are not included in a contact that we follow. So and so far as I know, we have 30% of the new cases or vice versa that are not included in contact we follow. So that means the transmission chain is not complete yet, but I know there are work on that are ongoing. For example, the sequencing, the genomic sequencing is ongoing. So we have a publication that was issued last week by Placide actually, so that show how the cases are being linked in this outbreak. So I think we still have challenges. This is one of the challenges we still have in this outbreak going forward, is to make sure that all the new cases come from contact or from followed contact. Over.
Yeah, thanks a lot. There is one question. I don't know who is going to respond to this question, but I think it's a real question. Why do healthcare workers in Ituri not have access to MVP 134 outside of the study protocol, whereas Americans evacuated to Germany do? I can start on that.
This is a very it's a very good question, but it's not true because the healthcare worker. When the healthcare work, we had some cases of healthcare worker, we did not have drug on site. The few healthcare workers that were infected and unfortunately some died, we did not have the drug on site. And you can remember that even the first American, we evacuated the colleague to Germany without taking any drug because we did not have it on site. So then when we received the drug, I remember that even we had an accident, one health worker received directly the Remdesivir because we did not have MVP 134 on site. And now when we started the partner trial, we brought both, so MVP and Remdesivir. So this is why it was possible to provide it to the American who was confirmed, because we need to mention that our colleague from Alima was not confirmed. He was accidentally exposed, but he remained asymptomatic and until the end of the treatment, and until now he did not show any sign or symptoms. So yeah, I think all health workers here, Congolese and other coming from outside, they will be treated the same in case of exposure, and in case of for example, if one of them or one of us is confirmed with the Ebola virus. Over.
Yeah, thanks, Placide. And I wanted to say that you know that in France we had a case. Finally, he didn't have NBP 134, but we discussed with partners. We wanted to include the patient in the clinical trial. And at that time, it was not possible. It was not even started in DRC. So I think that what you're saying is very important. First of all, we should not use experimental drugs in non-RCTs. We should push everyone to use in Europe, in US, and everywhere in RCTs. When there is not any RCTs available, maybe things are different. But I wanted you to know, as Amanda and Libby are saying, we are currently trying to put Europe and the US in partners, and also the same thing for Ebopep, so that ethically we do the same thing everywhere. And I really think that your comment is very important. So thanks a lot. I will
just one quick comment. Yes, now just 1 minute, just to explain that we have many health workers here, and we see that the exposition is almost the same. You know, regardless Congolese or other colleagues coming from outside the country or the continent. So and when we discussed with colleagues from American. Because of our American citizen contaminated, I mentioned that we have the RCT right now. We know that we may need also some drugs for compassionate use, mostly for where we don't have the RCT activated. So this is also, you know, a way to tell all of us that if we can, we will need to provide a few doses that should be prepositioned in DRC, and that can be used for compassionate use in a place where we are not able to kind of have the RCT in place. Over.
Okay, thanks a lot, Placide. I think very important. Now, we are going now to go to the Ebopep trial. So, and I will show you a figure to show you when we started this trial since when the epidemic was declared. While Ebopep is, maybe I'm just going to ask Steve Auka, if you agree, before the trials to give a speech. As I told you before he joined, Steve Auka, who is a physician in INRB, that we know for a while, with whom we work tremendously, has been appointed by the President of the Republic of DRC, outbreak manager for Ministry of Health for this epidemic for DRC. So I just wanted to just give him the floor, few minutes for his remarks. And thanks a lot, Steve, to being present with us. Steve.
Yeah, thank you very much, Yazdan, for the floor. Hello to everybody. I would like just to say a few things. You know, basically, I'm a researcher, and I know how the research can help the outbreak management. So, given the extent of this outbreak and the complexity that you may already know, it is important that the research on especially countermeasures should be, you know, reinforced to support and to address some key questions that we have, including the treatment, vaccines, and also prophylactic treatment. So I do hope that people who are here, we are committed to address this question, will be fully committed to join us to fight this, you know, disease, special disease in outbreak. So thank you very much for hearing me. Thank you.
Thanks a lot, Steve, and thanks also for your commitment, all your efforts, and your help. So, and for being present. So, Placide, the floor is yours. And we have hot discussions with Jake, you will see in the chat. So please, Placide, I saw that Gypsy put your slide, so thanks a lot, Gypsy. And news from EboPep.
Yeah, thank you for the floor, Lesda. I also share my slide with you. So I'm going to present on the EboPep study, which is Ebola Zaire post-exposure prophylaxis, preparedness and efficacy evaluation during outbreak in Central and West Africa. As you can see, it's a consortium of several organizations from Africa and also outside Africa. So it's a kind of EVD prevention in contact. So we are targeting high-risk contact, as you can see. And the high-risk contact, the way that we define it here is, kind of a direct contact with a person with a PCR confirmed of Bundibugyo, with diarrhea, vomiting, and external bleeding. So mostly people who are presenting wet symptoms, or if we are in contact, direct contact with the body fluid, and also direct contact with the dead body of a person which is who is confirmed or probable of Bundibugyo, and also if we have a case of like a needle stick with syringe contaminated by the blood of a person with confirmed or probable Bundibugyo. So here we are thinking, we are targeting, as I say, a high risk contact, and the way that we are thinking to do that is mostly to see how we can prevent high risk contact to develop an infection. So the context of this PEP came from different, I can say, observation. First, the fact that we know in the previous outbreak, when there is an outbreak of Ebola caused by the strain Zaire, we provide the vaccine to contact of confirmed cases and contact of contact. But what we observed was that people who are defined as high-risk contact, even when they are vaccinated, some of them can develop the disease. And also what we know is that according to the observation and published in the PREVAC study, they can see that the antibody, the vaccine do not provide kind of real antibody before day 10. So then we conducted also a kind of observational study during the 10th Ebola outbreak, and where we identify high-risk contact, non-vaccinated, and they receive MAB 114 and Regeneron, and we follow them up during 14 days, and no one developed the disease. But this was an observational study, and it was difficult to kind of draw a conclusion regarding the efficiency of this
l'efficacité de cette procédure. Et c'est pour cela que nous avons décidé de travailler ensemble, de créer ce consortium appelé EboPep afin
that we can use when there is an outbreak to see how we can, in a very good way, evaluate this EboPep procedure. So here you can see the number of countries and also institutions involved in this consortium. And so as you can see, the EboPep is really a key component for a comprehensive research response of the Bundibugyo outbreak. So it fits really well, among all the different platform studies that we are putting in place here, as we are facing a strain which does not have vaccine and approved treatment. So we think that working to build this platform to target the patient, to also prevent the infection among all contact, but also targeting high-risk contact is really important. And the protocol that we developed together with all different colleagues, and this it's a kind of a master protocol, and the sponsor of this study is INRB and sponsored by INRS. And there is a master protocol which includes kind of all different level of exposure of all different population. And inside or the annex of this master protocol, we have sub-protocol that can introduce some specificity. And as you can see, with the master protocol, we have all level of contact, which target all different strain and multi-country and multi-outbreak. And the sub-protocol is a bit specific, can be specific for the country, for the level of contact, study population, and also can be specific also for the different intervention. And for the EBOPEP study that we are conducting now is a double-blind, placebo-controlled superiority trial with 2 parallel arms. And we are evaluating the safety and efficacy of obeldesivir versus placebo in preventing symptomatic Bundibugyo virus disease in participants with high-risk contact. And so we will randomize one by one between the 2 arms. And we are planning to conduct the study in 2 countries, in DRC and Uganda. Those are the 2 recently affected countries. And the way that we would like to conduct this study is it's a flexible, where we are providing a oral medication that can be followed by adult administration, and there will be like twice a day during 10 days. And so the obeldesivir is almost the same activity as remdesivir, but with a mechanism action the same with remdesivir, and no meaningful relevant drug interaction. And it's kind of this drug has the advantage to be administered by oral, and also there is no cold chain needed for the storage, and it can be easily combined or with other previously approved Ebola therapeutics, and it's recommended by WHO. And you can see the figure on how the drug can move from different compartments of the body and also reach the target tissue compared with remdesivir. And regarding the population of the study, we are planning to include non-pregnant adults with high-risk contact within 5 days, and with no sign and symptom, participant with no sign and or symptom of Bundibugyo disease. And we will not include participants with previous history of confirmed filovirus disease within the last 5 years, patient less than 12 years old, pregnant or breastfeeding woman, and also participant who present with decompensated cirrhosis or renal injury. And so this is, I can say, the schema that show how the study will be conducted, where we start with the high-risk contact. There will be follow up to day 42, but we'll get the drug during 10 days. there is some flexibility here depending on the availability of the contact, so they can come every day to get the drug at EboPep site, or we can use the advisor community to see them every day to provide the drug, but they will need they can come back or they need to come back on day 1, day 5, and day 10 for and day 21 for visit and blood collection. But it's also possible to kind of do a home visit. So we make it a bit flexible, and we think that this will help us also to kind of accommodate the schedule of the different contact that or participant that would be included. And so inside this protocol, we have this sub-protocol regarding, the use of remdesivir among pregnant and breastfeeding women, and also children under 12 years old. And this population will receive a compassionate use of remdesivir when they are identified, because of the remdesivir, the remdesivir have a kind of embryotoxicity data, but not published by Gilead, and there is no pediatric formulation available for the moment. And so what we achieve, as you can see here, we move from the ethical approval, we receive the drugs, we activate the site, and yesterday we include our first participant, and there are 2 potential participants that can be who can be recruited today. So this is, I think, now we receive funding since 2023. As you can see, most of the funding that we received were used to prepare the different sites, as we did not know where the outbreak will occur. And so now, with the remaining fund, we are working to conduct a study in Bunia, but we need additional funds, about 13 million. So we are working to see how we can get additional funds. And there are other sources of funds like the BPI France, and we have a commitment from Africa CDC for 1 million, and we'll discuss with Yasna after this meeting to see how we will move with this funding that will help also to kind of support the site. And there are also other source of funding that we are seeking, like from the Gates Foundation and the Wellcome Trust. So thank you so much for your attention. I'm happy to take questions.
Yeah, no, thanks a lot, Placide. I want really to, we have these 2 trials with less than 2 months started. I think we should really thank all the team of INRB and INSP, all the DRC colleagues, but also WHO Africa CDC colleagues. Of course, we should even get better, but I think it was important to share with you this today. If Placide, if you agree, I wanted to also give the next talk of vaccine by Russia Bonavitz from CEPI, and then we will do, before Eddie's talk, question and answers, okay? So that it make it a little bit more. We have a lot of questions already on vaccine, a number of questions on very important on vaccine and post-exposure. We will keep it. I have them. Can you stop sharing so that actually Russia can? Thanks a lot, and thank you, Gypsy. Raquel, are you there? although I cannot see Rebecca.
Rachel is there.
Oh, she's there. Okay. Thanks a lot, Rachel. Thank you. So, and then we would do the question. We can see your slides. They are not on. Yeah. And also, please, Vazi and Mosoka, do not hesitate to jump in if you think that I talk too much, okay?
No, it's all good, Yazdan. Thank you.
Over, Rachel, please.
Thank you, Yazdan. Hello, everybody. Thank you for the opportunity to talk. Yes, I see a number of questions, but I think I'll proceed with the discussion and then we can have the Q&A as Yazdan has said. So I'm happy to present here on behalf of CEPI as the Filovirus program lead today. And, you know, as given instructions by the organizers, I'm here to talk about the research strategy, which will focus on the CEPI-funded BDBV-specific vaccine candidates, which I want to be clear, these are not the only vaccine candidates that are in development globally, but are the ones that I can speak to as receiving CEPI funding. And, you know, one of the questions really is, knowing that there are either licensed or investigational stage vaccines for other filovirus species, such as Ebola or Sudan virus, you know, the focusing on BDBV specific is that really these are most likely to elicit the specific binding antibodies and the neutralization for this species. The second part of the talk will focus on the current limited evidence regarding cross-reactive antibodies and the potential for cross-protection with those other licensed or clinical stage filovirus vaccines. And we'll do a brief review of the evidence and a review of the CEPI call for the proposal. And kind of from a larger, you know, lens at first, just thinking about CEPI's strategic approach, what I'm really going to be focusing on are these first 3 boxes from left to right. So rapid response platform vaccines, RVSV platform vaccines, and then evaluation of cross-reactive antibodies, really by leveraging serological studies of existing filovirus vaccines for the potential for either cross-neutralization or cross-protection. And then this question of whether clinical trials are to be considered if those results are promising. You can also see, and I'm not positioned to speak about this today, the question about post-exposure prophylaxis and therapeutics and what support can be provided there, as well as underlying CEPI's entire strategic approaches, enabling activities such as enabling science, you know, assays, operational support, partner coordination, and then certainly the focus on equitable access when thinking about either post-trial commitments and then ultimately the timely and affordable supply of accessible product for outbreak response. But we'll focus first on the 2 green boxes and then the first yellow box. And so again, in early June, CEPI announced these 4 vaccine candidates for Bundi-specific investigational vaccines, really arising out of longstanding CEPI partners with proven capabilities. And again, these are not the only candidates that are out there, but what I can speak to is receiving CEPI support. Going down the list, really, you know, there are 2 RVSV constructs with 2 different developers noted in parentheses here, which these constructs in the RVSV, which is the platform used for the licensed Ervebo vaccine against Ebola species, has some preliminary NHP data, which a caveat, NHP challenge model in this species, unlike the other filovirus species, is not a 100% lethal model. But regardless, the NHP data is in existence already for these RVSV constructs. And you can see the protection that is demonstrated by these constructs in a pre-master virus seed sort of situation with the key references cited here. However, there's not yet any BDBV clinical data, which will be consistent down this table for all of the vaccine candidates at the moment, though, as was noted earlier, there has been initiation of recruitment into a phase 1 study this week for the ChAdOx1 candidate at the bottom of the table. Moving down, there's an mRNA BDBV construct, which does not currently have any NHP data for BDBV, but mRNA constructs with Ebola, Sudan, and Marburg GPs, you know, there is preclinical data in guinea pigs that is published and other preclinical data that is pending publication with the references there. There's also additional small animal preclinical immunogenicity that has been obtained as vaccine development has been accelerated. But again, emphasizing that there's not yet any BDBV clinical data until phase 1 trials can be initiated. And then last on the list is the ChAdOx1 BDBV I guess important to note, similar to RVSV, which is the platform used for licensed vaccines with other filovirus species, certainly the mRNA platform from Moderna is used for licensed COVID, RVSV, and flu COVID combo vaccines. Then going down to the last one on the list, in terms of the ChAdOx BDBV vaccine candidate, again, no NHP data currently for BDBV. There is preclinical and clinical data, however, with EBOV and Sudan GP in this construct with references cited. And adenovirus, or this platform, is the platform used for a licensed COVID vaccine. So, there is extensive safety data with these platforms that these vaccine candidates are leveraging, and definitely have demonstrated preclinical, efficacy against other filovirus species in these vaccine platforms. One other thing just to mention is that for the ChAdOx vaccine, in the last row, there is currently unpublished data in preparation that demonstrated comparable efficacy of a monovalent Sudan ChAdOx with Sudan RVSV vaccine in a ferret challenge, which is in prep but not noted on the slide here. And then, as I'm sure folks on the line are aware, CEPI did launch 2 complementary CFPs to accelerate development of vaccines by delivering outbreak-ready vaccine candidates in the near term, and then also, supporting evidence generation for vaccine R&D decision-making. CFP, the first CFP, or the accelerated development of additional early-stage vaccine candidates. This is included here for comprehensiveness. This call closed in the middle of June. Selection process is still underway, and it's included here for comprehensiveness, but is not really going to be the focus of the rest of my time today. What I'll really be focusing on is the second CFP that was launched on June 5th and closed to June 26th, which is the generation of evidence to support vaccine R&D decision-making. This is divided into 2 really distinct work streams. The first work stream being epidemiology, modeling, and analytics. Just to say here, this was providing support for epi modeling or analytic efforts that would enable, planning of clinical trials, enable that tranche of kind of enabling science research, but I won't be speaking about that further today. What we will be speaking about is the second work stream under this, which was the immunological and clinical assessment of potential cross-reactive antibodies induced by those investigational or licensed vaccines against other species in humans. And so just to say here, the selection process for this CFP is ongoing, but this is really kind of the pillars underlying the questions or the reasons for this CFP, and kind of this question about what can we determine about cross-protection. Certainly don't need to explain the first box here, but that there is no BDBV vaccine that currently exists, though there has been accelerated development and even, phase 1s beginning. However, cross-protection is unknown. And so as noted in May 2026 with the WHO tag CVP, there was limited evidence that EBOV, Sudan vaccines, together, prime boost and in isolation, protect against BDBV, NHP, sorry, that's a typo, NHP challenge. And there's limited human cross-reactive antibodies. And even those limited studies at the moment with cross-reactive antibodies, don't necessarily provide evidence of cross-neutralization, which would also be a corollary to this cross-protection question. However, considering serologic samples that exist on consented EBOV or Sudan sample sets from previous clinical trials, can deliver results in less than 2 months. And that was one of the requirements or criteria for the CFP, is that applicants are able to generate data within that 2-month time span. And really this would leverage prior investments in existing EBOV and Sudan clinical vaccines that could potentially be repurposed for near-term BDBV protection, I think, if this question is more fully assessed before going straight to clinical trials. And then certainly timeline considerations for the overall pipeline. Certainly, while there's been accelerated development of the early stage BDBV specific candidates, there will be time needed to reach phase 2, phase 3, and wondering if any of this data generated for the serological studies may be able to offer a bridge in that meantime. And certainly in the future would inform a pan-vilo strategy, which really, a pan-filovirus antigen design for future vaccines is scientifically justified. And so, the justification is there are already GMP grade existing EBOV and Sudan vaccines, whether in stockpiles or in investigational kind of supply. and then answering these questions on serology with the potential for near-term protection. Again, this selection process is ongoing, and I can't speak more to specifics about that at the moment, but the expectation is that the data generated from these selected proposals, especially given this time span, is expected to be reviewed, including by WHO SAGE, in the very near term and before next steps. And this is a slide that probably folks have seen before, is a little bit updated because certainly, from the pre-vac up group, you can see the paper from 2026 that has been added here in terms of looking at cross-reactivity or any sort of cross-protection evidence. But I think what can be appreciated is that this is incredibly limited across the different vaccine platforms and or the different developers. So certainly with Ervebo or, an RVSV EBOV vaccine, partial protection against BDBV challenge in macaque models, which wasn't sterile protection, and animals had viremia and were symptomatic. And I think previous CORCs have gone specifically into this data, but with the time constraints today, we're keeping it at this high level summary. There was the paper from 6 Ervebo recipients in a phase 1 study, which did demonstrate cross-reactive monoclonal antibodies to BDBV, but again, it's 6 individuals that have been published. And then Ervebo induced detectable but quantitatively reduced cross-reactive antibodies against BDBV coming from out of the PREVAC study group, in a Luminex multiplex assay, which is really also difficult to assess or know the protection, which is really this question that I think is the crux of the question here. And so a lot of these studies and even these further studies further down on the table really is this call for standardization and the use of neutralizing assays. It doesn't mean that there isn't also a wealth of unpublished data from additional groups, but hopefully as some of that data becomes published, and then certainly also with the call for proposal that CEPI is conducting right now, that this comes to perhaps better standardization of assays and the use of neutralizing assays to better answer this question, separate just from quantitative assessment of cross-reactive antibodies. Certainly, there is also some data from one study of samples from vaccine recipients from the AD26 and MVA regimen, for which the license has currently been withdrawn from the EMA, which showed lower absent ELISA or neutralizing titers against BDBV. And similarly, from the same PREVAC study, it induced detectable but quantitatively reduced cross-reactive antibodies. And then with the ChAdOx bivalent construct with EBOV and Sudan glycoproteins, there's no published data, but there is published data on lack of Sudan protection in NHPs with that bivalent construct, certainly. And then those are, vaccines that have been licensed or were licensed. And if we move into other vaccines that are have investigational, clinical grade vaccines available, you can see the minimal evidence here as well, with a chimp adeno 3 Sudan vaccine with no published data on any BDBV immunogenicity or protection in preclinically or clinically. RVSV Sudan vaccine, same thing as the chimp adeno above. And then, this question about an RVSV prime boost with Sudan and EBOV vaccines showed improved protection against BDBV and macaques, but when the vaccines were co-administered, meaning not along the prime boost schedule, but at the same time, there was no protection. So there's really confounding data here and very small numbers. And again, while these vaccines exist as clinical grade, in the study, lab produced material was used. So it's not, exactly, the same as the clinical grade material that is either licensed or available for investigational use. And I think that really brings me to the end. It might have been quick, but trying to preserve time, Yasin, and I think we can pause here now for any questions. I'll stop sharing, and I'm sure there are many, many, many more questions in the chat.
Yeah, thanks a lot, Rachel. This was great, and thanks for your effort. So there are a lot of questions. So maybe we will start with EboPep and then vaccine. We will take 11 minutes for question. So first for EboPep and Placide and Gypsy. So one question comes. So we have Billy, Jake, and so Jake Dunning, William Fisher, and from Lauren Sower, saying that if they have a healthcare worker with exposure, why randomize it in RCT? So I leave Placide respond, but if I can, so you think without a trial that this drug is efficacious, right? As we thought that tecovirimet was efficacious, we gave it for 3 years to everyone, but it was not efficacious. As we thought, some people thought that hydroxychloroquine was efficacious and it was not efficacious. So I'm a little bit surprised, but I give the floor to Placide and to Gypsy to respond, probably.
No, I will go the same direction, guys, because we don't know if it's efficacious. So this is why we think that we can randomize. But this is a, it's a volunteer study. So meaning that we propose to the healthcare worker to be part of the study because we don't know if it's efficacious or not. So this is the reason why we think that we should also randomize.
Yeah, and I think this is a discussion we have for now years and years and years. So, but anyway, Benzi, you want to say something, please?
No, just to agree. We don't have any proof that we have a safe and efficacious intervention here. So the way to generate evidence is to generate evidence. And we cannot generate evidence unless we do high quality research. The best quality evidence is going to be through RCTs. So there seems to be some kind of supposition of some evidence that doesn't exist. That's the whole reason why we're doing the clinical trials. If we had evidence that they are safe and efficacious, then it wouldn't be appropriate to do the clinical trial. So if I had that exposure, I would certainly want to be randomized into partners if I could. It's much more difficult if you're in a situation where the clinical trial is not set up. Thank you.
Thank you. And I think that we are not here to do popular things because we are scientists and not politicians. So maybe another question is from Mireille Ramon on, we saw Marie's data. Those with the highest mortality are children, basically. And we cannot do this study in children. So I don't know if Placide, you want to say why we cannot do and what we do. I think that is a question that is asked that maybe you can respond.
Yes, no. So as I explained, we are using orbedicivir, and with orbedicivir, we don't have the pediatric formulation yet with Gilead. And because of some data not published, but which showed some embryo toxicity in pregnant women and also in pregnant women. So we decided to provide a kind of compassionate use for those who cannot be randomized because we cannot use orbedicivir for those patients, like children under 12 and also pregnant women and breastfeeding women.
Yeah, thanks. And the idea from INRB and colleagues and some have been, the discussion have been with Gilead to try to have a phase 1 study looking at the safety. Because I completely agree with you that the fact that we have such a high mortality, if it works, it's important to be used also in children. Then there is one other, I don't know if Placide, you can respond to this question, is a statistician response, so you or Gypsy. It is from Daniela Mano from the UK. I noticed that EboPep is individual randomized rather than using a ring or cluster. Could you say a bit about the thinking? regarding this, and why not ring? So it is a difficult question. I don't know if you can respond. If not, I saw that Laura is
very,
I knew.
very difficult.
Yeah, I don't know if Laura can speak.
I may
Laura,
refer to our statistician.
yes,
to
exactly. I
give
knew,
the
but you are
answer.
also a statistician, no?
No.
So so I don't know if not, we will respond Daniela by email. I don't know if Laura, I saw that Laura was on the, if you can respond, Laura or Ira, maybe that would be great. Now, maybe some.
Yeah, but maybe well, we can discuss with Daniela even after the meeting,
Okay,
because
yes.
it's good to understand different data.
Now, maybe some questions, Rachel, regarding the vaccine. One question is coming that I will start maybe with this one, is regarding the sequence and the fact that I cannot find again the question, but that the sequence is a little bit different from the historical sequence, and does that matters? And I will find out, but probably you saw that question, yeah?
Yes, there's a lot of questions specific to the sequence, and I think I will
Yes.
preface it by saying I am not the developer, right? It may not be at liberty to disclose confidential information from the developers, but I see Joan is in here, and perhaps others are representatives from the developers are here and maybe directly answering some of these questions in the thread. I do want to confirm from the Moderna mRNA construct that their strain is matched for 2026, and I'm at liberty to share that. And I see that there are additional answers in here, but and directed now specifically to some of the individuals. So maybe we can hold on that one and then see how folks are answering in the thread just for now, Yazdin, and then we can circle back and certainly follow up offline.
Okay, so then that, and then, and I see that actually Ira has responded to the Daniela's email. His question, individual randomization is much more efficient than cluster randomization, requiring a much smaller sample size. So you have a response, but I didn't see it, sorry. And then one other question, or the, so yeah, that was it's a bunch of questions regarding sequences, and also the fact that the antigenic shift, whether it's anticipated, so that we would see then. One other question, I think, is whether we can, through observational studies, look at the efficacy of Erbevo. So not to do actually a study, but I know that MSF was going to conduct a case control study. I don't know if Raquel, you know about that, of among the cases, see those who have been vaccinated and those who are not being vaccinated. So I don't know if you want to answer on that.
Sorry, yes, no, is that for me?
Erbevo.
Yes, SEPI's aware of these MSF plans, but we're not directly involved or directly supporting. So I think probably I can leave it there, unless there's anybody else on the line that would like to specifically answer that question.
And if they want, I know that because I talked to them just before. People are MSF are on the line, so I knew that they wanted to do a case control. So it would be good if they can put on the chat for everyone. We have also from Logan Elliott. From NMEL Winnipeg, saying that they have some preliminary data on VSP, mentioning that it could actually protect against BDBV at 28 days. But as you said, I think, Raquel, those data are not strong. Maybe you can say something regarding that, actually. Because there are a lot of questions regarding BDBV actually, and Erbevo.
Yeah, and thank you, Miles, for linking to the evidence map, which probably is a much more comprehensive review than I could cover really in the short amount of time here. But I think in general, the evidence base, and I understand that binding antibodies are the basis of licensure and the basis of, have been an endpoint that's used in licensure, but certainly with the example of Ervivo, there was an efficacy trial prior, right, with the binding antibodies. So the question is, are the binding antibodies that are induced at relatively lower quantity and not completely uniform across the board, the same thing as cross-neutralizing or providing the same sort of efficacy? And I think we can't answer those questions with the serologic evidence that we have right now. Certainly appreciate the comments that everybody's making, though, about the importance of binding antibodies and how that has been,
Yes.
used previously. But I think in the absence of cross-neutralization, we are really limited to say anything about cross-protection. And so the hope is with this call for proposal and assessing cross-neutralizing antibodies and maybe getting a larger body of evidence, we may be able to better provide additional data, certainly with WHO SAGE as one of those stakeholders there, to review this data in totality before, potentially transitioning resources to a randomized clinical trial, where you'd also have to ask yourself, is the endpoint for a randomized clinical trial of licensed or investigational vaccines an immunological endpoint or an efficacy endpoint, right?
Yeah.
Given the concurrence as well of advancing BDBV specific candidates. Sorry, Yazdan.
No, exactly. So, no, I think that is very important. And, there are also questions, this is the other side, regarding whether there are concerns regarding impaired immunity from anti-vector immunity in people who have previously received ChAdOx. Is there a concern, preclinical evidence regarding immune imprinting if having received Ervivo? So there are lots of questions right there.
A number of questions there, and certainly about anti-vector immunity, which I think is less of a concern, but certainly also important questions that I don't think any of our available data that we have now answers. Sorry, the chat is just continuing to.
Yes, it is. It's getting mad, but that is good. We
Yes.
are not going to, and there, of course, we are not going to be able. So I think discussion should go. We will try, to be honest, to respond to the question in the chat. I just also to mention that Libby, so Elizabeth Eggs is saying that Gilead is working on the pediatric formulation, and we have discussion with Penta, WHO, and I have to also mention NIH. And I think that I'm going to try to stop here if you're okay, because if not, we're going to. I will see if we have time to. I will look, because I don't have the time to do both things, but to come back. But thanks a lot, Russia, and thanks a lot, actually, Vazi and Gypsy. I have to say also that Marie is not here today, Marie Jaspar, but thanks her also for all the reports. And what we're going to do now is going to do the next topic, which is diagnostics. And we did some working group of diagnostics, because we always talk less about diagnostics. We talk it's important. And it's a pleasure for me to invite Eddie Luzamaki from INRB to give us a review of what's going on diagnostics. Merci beaucoup, Eddie.
Thanks a lot for the floor. Just sharing my screen. I hope that you can see it.
Yeah, we can see it perfectly, but we would like that DRC go a little bit further in the World Cup, to be honest. Football World Cup. No, I'm just saying, because I see the flag, so I was just saying. But anyway, thanks a lot.
Okay. Yeah, so we'll briefly go over the diagnostic and the research and development updates regarding that. And as Yazdan said, we've been doing some work within our group. And as you can see, that this is tightly related to the previous discussions that has been undertaken by the previous presenters, and thanks for the insightful presentation. So we will start by going through the diagnostic, how we came to the outbreak declaration, because keeping in mind some historical overview is still important, and then we will go through the diagnostic overview and give some updates on the research and development. As we know, we are talking about filoviruses and especially ortho-Ebola viruses. And the current outbreak we are experiencing is caused by the Bundibugyo virus. And we knew before 2026 that we had 2 historical outbreaks, and these last ones started in the eastern part of the country, as many of us are aware. And for a diagnostic, I wanted to spend some time here because when we received the alert, the samples were tested GeneXpert negative and then shipped to the INRB, where we did in parallel testing using Altona kits, which target Ebola viruses at the genus scale. And also we used a point of care, which is the RADI one, which also targets Ebola genus or ortho-Ebola viruses. And finally, we also used the BioFire platform, which also targets ortho-Ebola viruses. So without defining which species is causing the disease. And we understand that subsequently we had to perform the sequencing to be able to better characterize the strain. And we've been able to define that it was a new spillover, which was occurring both in DRC and Uganda, and responsible of the new outbreak we were facing. And rapidly, the diagnostic response was made up. You could see that Placid is deployed in the field with a team and many equipments, both point of care and open PCR thermocyclers have been also deployed in Bunia, which is operational. We also trained a local team so that they could perform both the PCR, and also a mobile sequencing lab has been deployed in Bunia and also in Goma right now. So there are a lot of more than 10 mobile labs that have been deployed and which are operational for testing right now in DRC for the outbreak. So where are we with the diagnostic? We know that there are a lot of approaches that can be used. We know that the gold standard is the RT-PCR for the Ebola, which provides high sensitivity and specificity, even though it's requiring some training, and there are also some requirements regarding the sample transport, and the infrastructure also needs to be well defined. While the point of care, which uses most of the time cartridges, can easily be decentralized, as we are doing right now with an outbreak setting, and deployed to the field. But many of the time these are closed system, and also it is then platform dependent, and also there is a question of costs. There are a lot of other strategies which are being put in place and still being tested, like the isothermal one, the question of RDTs, which could allow a deployment and which is faster, but still has concerns regarding the sensitivity. The sequencing, which is ongoing, and the serological as well, which are requested. And we could see that most of the tested, so we could make an inventory of 27 commercial tests, but we can see on this map that there is a huge discrepancy because there are no local manufacturer identified in Africa, and most of the outbreaks occur in Africa. So this is a very huge question that needs to bring attention. And also we can see that most of the tests which are being developed right now are RT-PCR tests and technologies. So many commercial kits are approved either by WHO or Africa CDC are available right now, and even also for the immunoassays, the list is there. So I will just go rapidly through this. But we also know that many other tests which are still for research use only are available, and some of them can also be used for the research purposes. And the same for the immunoassays. So many of those tests are now being also validated so that the performances can be easily evaluated regarding the current strain we are facing of Bundibugyo virus. And other tests are still in development. So this is really a field in which most of the efforts are needed because the medical countermeasures are not yet ready. So diagnostic is like the eye of the surveillance to help to identify the transmission chains and to break them rapidly. I will end with this last slide, which summarizes the research and development roadmap. So far, we know that tools exist. Molecular, antigen, serology, sequencing options are either available or in development. But there are a lot of barriers, like the validations, informations on the sensitivity and specificity, either in a lab with good infrastructure setting or in field setting. We really need to assess that. And there is also some other parameters on which the performance of those tests depends, like the access or the supply of other reagents. Therefore, innovation really is requested and weighted. So we know that that innovation should focus less on adding isolated tests with closed platforms and more on closing the gap between available technologies and outbreak-ready diagnostic system. And one of the aspects which should be targeted by those innovations are the samples to be used for the diagnostic, the multiplex approaches, which can help to target all the Ebola or filoviruses viruses for better surveillance, both for molecular and serology, the genomic sequencing to make it broader but still more sensitive, and we have not to forget the RDTs, because for Bundibugyo, till now, we have no RDTs deployed on field, while it has been described in the first presentation that we have a lot of community deaths. So this should definitely help. We have not to forget that Ebola is a zoonotic disease. So handling this using a One Health approach and considering also the other potential hosts should be ideal, even for the diagnostic. With this, I will just thank all the colleagues from INRS, from INRB, all the partners who are still supporting for the outbreak. Thank you for your attention.
Yeah, thanks a lot for your presentation. As usual, Eddie, perfect presentation. Thanks a lot. So one question is that I can see that Sylvie Briand is here. Sylvie, can you stay until the end for the conclusion, or you should leave? If not, if you can stay, we wait for the end. If not, you can go on. Just
No, I'm going to stay for the end. It's very interesting conversation. So
okay.
thank you very much for allowing me to stay until the end. Thank you.
Okay, thank you. Thanks a lot. Thank you, Sylvie.
Yes, Dan, you're muted.
Thanks, Nadine. No, I was just saying that we are going to finish with priorities, but maybe just few. I'm going to see if there are questions for Eddie. But first of all, regarding the sequence, maybe there's several responses, but those who don't read the chat from John Henriks from IAVI is that the use of the order seconds allows for the direct extrapolation from the ENHP protection data. So that is why, and it's the fact that the sequences are nearly identical between 2012 and 2026. So that is, I think, important. The fact that there are case control studies going on. There are 300,000 people in North Kivu and Ituri that are vaccinated. And what Ira is saying that I didn't know is that INRB is conducting a text negative design for cross protection for Vivo. And finally, yeah, just this is a very passionate discussion about clinical trials. And I'm sorry, Jake, I didn't want to paint anyone. This is a fair discussion. I agree with you. It's passion discussion. I'm always clinician, and when I am a clinician, I think different thing as a scientist. And I think what Billy is saying, which is important, is that the risk benefit when there is a needle stick injury is very important. But so let's continue to discuss, although we are for RCT, but we're not painting anyone. Sorry for that. And Vazi, you want to say something? I saw that you opened.
No, I don't.
Okay. And then just maybe one or two questions for you, Eddie. What measures is One Health implementing among healthy unaffected people? Or I don't know if you know that.
Sorry, I did. Yeah.
What measures are in place in DRC to limit people's contact with bats? I don't know if maybe it's, I don't know if Steve is he's still here, but, or maybe plus.
Yeah, I think they can chime in and add in, but what I know so far, so that there were meetings, especially after the mpox outbreaks we have been dealing with since 2022, 2023, 2024. There has been a lot of efforts to try to implement One Health approach using especially animal surveillance and also environmental surveillance. So there are still efforts which are trying to be built. We know that DRC is a very big country and also low and middle income setting. So there are still a lot to do within that. But to limit contact with bats, it's a bit difficult. There has been some, with some projects, some educations or some sensibilization, but at some area, yeah, people, it's especially bats, people are still interacting. So these are still things we need to look in deeply. Over.
Thank you. And one final, and then there's another, if you can respond it, but it is from it is from Sophie Trouillet from Lyon. So what I think it's a very interesting question. What is an acceptable price for a diagnostic assay? Just say one range.
Yeah, if we have to talk about range, and especially from a DRC perspective, because we must not forget that DRC is still among the low and middle income countries. So I will take between $1 and $20 or euro.
Yeah, so closer to $1 to than to 20, right?
Yeah, in that range.
Okay, thanks a lot. And now I'm going to present. So this is a collective work, of course, with WHO, with Africa CDC, and Dr. Musoka is going to give a few words at the end of the presentation. But the idea is that we have been working on filovirus research priorities with everyone. And the idea is to present you actually the first data here, and you will receive the document. So this is, I'm not going to do this, but this is just to give you the actually what has been done. So the beginning of the epidemic was actually, at least the declaration, sorry, was on May 14th and 15th. And this is what has been done. One thing that is here I have put is July 2nd, patient enrollment in partners, July 14th, the beginning of EBOPEP, and July 13th, the beginning of phase 1 vaccine trial of Chad, actually, of ChAdOx. So the question, of course, that we will ask ourselves all the time was whether this is enough or we should go faster. But this is just to show you. Then the second thing that is important for us is how we did this methodology. So all the CORCs have a roadmap. This is the roadmap, which has not yet published because it on filoviruses. So it's not on Bundibugyo, it's on filoviruses. But it was not published because we were still working on that. When the epidemic occurred, we adapted that to Bundibugyo. Then we tried through consultations with CORCs, but also with Glopidar, but also based on WHO tag recommendations to refine those priorities from the general roadmap. And then what we did actually, what we did is that those priorities were reviewed by WHO, Africa CDC, and international panels. We had a meeting, we discussed it. And what you're saying is that it's actually the result of all this. So first, roadmap, then refinement, then international consultation and presentation today. The prioritization criteria was the impact on patients' outcome and viral transmission, the urgency of generating evidence, to the potential to inform decision-making, the relevance to BDBV knowledge gaps, and the cross-cutting value. These are the priorities. So first, the natural history and pathogenesis transmission. Second, social and behavioral research. Third, diagnostics, genomic surveillance, and data systems. Fourth, supportive care and clinical management. Fifth, monoclonal antibodies. I'm not going to detail everything. You will have it in the document. Sixth, the antiviral and innovative therapies. Seventh, post-exposure prophylaxis. Eight, vaccines against BDBV. 9, recovered patients, survivors. There was a question. We are working on that. We try to put some discussion next week. And last, infection prevention and operational research. So these are the 10. What we try to do is to say how urgent they are for implementation. So we define different phase. Phase 1, 0 to 6 months. Phase 2, more than 6 months. Phase 3, post-outbreak. So you can see, for example, activating trials or characterizing what is BVD or strength, innovative approaches for what you heard today, established cohorts. These are those things that we should have very quickly. And there are some we will have, of course, we should implement, but the evaluation of, for example, PEP effectiveness is after 6 months, probably not before. The same things on optimization of management, of platforms and surveillance, etc. So I'm not going to do this in detail. You will have the slides, and we are currently reviewing with WHO and Africa CDC. You will have the document next week. I wanted here to actually thank you all the African CDC teams, WHO team, CORC team, INRS team, who have worked a lot. And one other, and I will stop sharing here, I will share now Dr. Musoka's slides, because we were also extremely, we want really to try to do, not do this with Africa CDC in silos, to do it together. So that's why what we have done was always in articulation with Africa CDC. And actually, Dr. Musoka, do you want to actually try to show everyone how we work closely together? Please, I can share your slides.
Thank you so much, Yazdan. So just before the outbreak, we had planned a meeting to bring together African scientists to look at arboviruses and filoviruses. So around this time, we reached out to Vasi and Yazdan and said we wanted to see if we could come up with an African context roadmap, but that would leverage upon the global roadmap. And so next slide. We met in Addis and we probably came up with these are the group of people we met. We came up with priorities for 10 priority areas in terms of Bundi. If we come to the next slide, we don't have the time, we'll share the slide with you. Next slide, please. So we basically choose 10 priority areas. And what we have here now showing is basically is those 10 priority areas and what are we doing now in terms of our research. If you see for sure, if for each of those areas, we are not working in isolation, we are working with other partners. If you look at the EPI protocol that we are developing together, and you see that we try to align most of this work with WHO work, and that's why you see in the last column, which we share on alignment. But basically, what we have here is the 10 priority we selected and how they align with what we are doing now and how they also align with the WHO fellow group. So we can share this. If you look at, for example, the survival studies, we are now working closely with Libby, also with Gilead and ENRB for us to develop a survival study. The same thing with the social behavior study. We are working with multiple partners. If you look at on vaccine and protection, we are working with MSF and other partners to see what we can do in terms of cross protection. But what this table shows is what we reflect as the African priority, the 10 priorities, and how do they align with the overall global priorities with WHO. And finally, when we share your slide, you see how they also speak to the kind of research protocol that we are developing. Thank you so much, Yazdan. You have all of that in the slide that will show how what the global is doing and what the African have agreed to do and how it aligns so that we all work together. Thank you and over.
Yeah, thanks a lot, Dr. Musoka. And do you want Vazi say now what is the next step? Because that is very important to inform you what we are going to. Please, Vazi.
I think it's Rina was going to speak, wasn't she, at this point?
Oh, yeah. Okay, we can do Rina and then you. Please, Rina. Yeah. I'm sorry. Go on, Rina. Because what we have a dashboard, and we thought that it will be good that Rina, and then I will give the floor to you. Sorry, please, Rina.
Go ahead, Rina, please.
Okay, so I'm just going to share my screen really quick, hopefully, and I will be very quick.
You don't have to hurry.
Don't hurry up.
I think it's important that the folks see.
We are going to be 5 minutes a little bit late, but that's fine. Please go on. No hurry.
Great. Sorry, my internet is a bit slow. So what we're trying to do as part of the Continental IMST research pillar is map out the BVD research landscape. So as the research expands across countries, institutions, we really need a coordinated approach to make sure we understand who's doing what, where research is occurring, and how we can all work together. So we need to make sure that multiple groups who may be planning similar studies without knowing each other about each other's works, that we can capture that. You know, it's difficult to identify who is conducting research, where the studies are taking place, and what questions are being addressed if we're not working on this. There's also opportunities for collaboration and protocol harmonization that may be missed, and opportunities for evidence gaps that may remain unaddressed if we don't have a system for recording. Also, we want to look across countries for research readiness and implementation status that might not be easily visible outside of the hotspots of the outbreak. So what we're trying to do is develop this tracker which supports the research coordination. It's a simple tracker. It's an Excel tool and also a Kobo tool that we will share with you. I'll share it in the chat, and also it'll be emailed to you as well. With the goal is just to create a comprehensive overview of all the ongoing and planned studies. We want to identify collaboration opportunities as well, make sure duplication of research activities is avoided. We want to improve visibility of research gaps and also consolidate research findings to inform the response, because the goal here is really to inform the response at this stage. So this is the sample tracker. It's a Kobo tool, like I said. It'll capture just a bit of information on the study that you're planning to do, how it applies to the priorities that were just shared right now. So we can also track by research priorities. It'll ask the country that you are conducting the research in, the region, the lead institution, and where you are, the status of that study and collaborators, and also whether it's already approved, it has ethics review. So it's very simple. It's mostly just limited writing and you just checking boxes when possible. And there's also an Excel file if you're more comfortable with that. Again, I'm going to share this in the chat and also email it out. And we really are encouraging you to fill out anything that you're planning to do, anything that you are currently doing, and also share with other researchers and collaborators that you know are planning or conducting research, so that we can really map this out and know what everybody is doing. So that is it.
Yeah, thanks
and
a lot.
this is share it as well.
Please finish. I thought you finished.
Oh, no, I'm finished. Thank you.
Yeah, thanks a lot, Rina. So you see, the idea is to link priorities to, and this, what Rina was presenting was very important. But maybe I give you now the floor, Vazi, to maybe give the next steps.
No, just briefly, I think what's important is that we really need to be clear that the medical countermeasures research is not more important than the other research. And I really don't want people to get that idea from this webinar. We deliberately wanted to have a whole other discussion, and we didn't want to squeeze. So the next steps really going beyond the high level priorities. And this is not to say that if research is not on the priority list, it shouldn't necessarily happen. But I do think it's important that we go through a process for the priorities, is then to identify where we have good quality protocols, where the scientific community and the ethics community is really comfortable that this is a good protocol to answer what is agreed to be a priority question. And then we as the scientific, as the community here, need to really do whatever we can to advance these priority protocols. So, I mean, I think some of the clear areas, but this is just not at all exhaustive, would include on the natural history. So we have some information on the clinical characterization, but I think we need more sampling to answer some additional questions around shedding and viral persistence and other questions around natural history. And for viral persistence, if we see that, we may need an interventional design to understand how to minimize that. And there are a number of other studies that we really need to look at. And once we have this map between, which is exactly what Rina is showing, I think hopefully within the next couple of weeks, we can get to the point where we're clear where we have priorities, that we have a protocol, and those protocols are not getting enough support to move forward. I mean, I think it's partly our role as WHO with Africa CDC and partners to reach out and try to advance these really priority protocols if for some reason they're not funded. So that's the way I see the next steps. Clearly on the social behavioral science, on community engagement, on any kind of research that we can do to improve the quality of the epidemiological information, where there's a lot more to do on advancing the diagnostic platforms. So there's a lot more beyond the treatment trial, beyond the prophylaxis trial, and beyond the vaccine trial. That's what I wanted to say. Back to you, Yazdan.
Yeah, thanks a lot, Vazi. And I just wanted to also to say what you said. So public health measures, very important. That's why we have another meeting next week, more on those issues. It's about 4 p.m. Geneva time, but we will confirm that through email. The other thing is that reservoir, it's not with that I saw that they say why reservoir is not, it is. You will have the text. I went very fast. You will have the text and you will see, Joe, that it is. Musoka, do you want to say a word before I give Sylvie to close, actually, on behalf of Africa CDC? If you want to say a word, I'm not sure that Musoka is hearing us. Dr. Musoka, we are.
No, thank you so much. Nothing else to say. Thank you so much for this. Thank you. Thanks to everyone. Thank you.
And I wanted also to thank everyone, WHO team, Nedi, Patrick, Ximena, all for a huge amount of effort to present this. My team also, Hervé, Vincent, and the other Vincent, and Melanie, because they work a lot. So thanks a lot. And Sylvie, and also all the people from Africa CDC. Of course, Dr. Bosoka, but also Dr. Placide with the other hat. So thanks a lot. And Sylvie, the floor is yours. And also Rina. Thanks a lot, Rina, because she has done a great job for helping us. Please, Sylvie, the floor is yours.
Thank you very much, Yazdan, and I'm really very fast because I suppose you have heard a lot of very interesting talks this afternoon. So I want just to close over just first, thank you all for your commitment, expertise, and openness over the course of this consultation. It was very interesting discussion. And as we close, I would like to begin by acknowledging the reality that brings us together, because behind every discussion or every scientific question are communities that have suffered loss, fear, and disruption. And this tragedy experienced by the affected population during outbreaks of Ebola, Bundibugyo, or other Ebola disease remind us that our work is not only a scientific endeavor, but also a human one. And we owe it to those on the front lines, patients, families, healthcare workers, researchers, responders, to transform the lessons learned into action and progress. And over these days, we have seen the power of collaboration, because no single institution, country, or disciplines can answer the very complex question that outbreaks present. And progress in science depends on partnership. Partnership that brings together clinical researchers, public health experts, communities, regulators, funders, policymakers. And this discussion have demonstrated that when knowledge is shared openly and expertise is combined, we move further and faster towards solutions that can save lives. And this was not the case before the Ebola outbreak in West Africa. So I'm very happy to see that things have changed and progress are made. We have also reaffirmed an important responsibility of the international community to build systems that enable research to be conducted rapidly, ethically, and effectively before, during, and after outbreaks. And scientific advances only achieve their full value when they translate into equitable access. So we must continue working to ensure that new diagnostics, therapeutics, and vaccines, once proven safe and efficacious, are available to the population that needs them most, regardless of geographies or resources. So looking ahead, I'm particularly encouraged by the role of the CORC as an innovative mechanism for coordination and collaboration. It's a new way of working that connects very different partners, but also allow us to align priorities, especially in research. And Yazdan has shown the research agenda that has been updated, but also it helps us to strengthen preparedness before the next emergencies and not look at each outbreak as a completely new challenge. I think we need to improve over time when we deal with these type of events. But the challenge before us is not only to respond successfully to the next outbreak, but also to institutionalize what works. And the successful processes or partnership and governance approaches data developed through efforts such as this consultation should be consolidated into a sustainable health architecture that can endure beyond individual outbreaks and funding cycle. So funders, please don't stop funding when the outbreak is over. And let us leave this meeting with a shared commitment to continue collaborating, to continue learning, and continue building the scientific and public health foundation that will protect future generations. So thank you very much for your engagement, dedication, leadership. Thank you, Yazdan. Special thanks to the very small WHO R&D team, and you mentioned them all, Yazdan, but also many thanks to all partners, Africa CDC and the CORC Filovirus partner and leadership. So I look forward to our continued collaboration, and thank you very much again for giving me the floor and for this consultation.
Yeah. Thanks a lot, Sylvie. I just wanted also to say one thing, because for both partners and EboPIP trials, we have also an Ugandan PI, which is Pauline Bikaka, and I think she should be here. I also Pauline Biakika, thanks a lot, because things are a little bit calmer and hopefully in Uganda, but she has helped us a lot on both. So thanks a lot, Pauline, also, and thanks a lot all the INRB, INSP, and all the people on the field. So if you are okay, we can now just stop this and we find you next week. Have a good weekend. Thank you.
Thank you. Thank you.
Recording stopped.